Hi, my name is Noor Naveen, and my experiment is BioAssay test for toxicity in seed growth. One third of the world's farmland is considered unusable because of high salinity in irrigation systems as well as the soil. If the land was able to host crops, it could aid with many major problems we deal with, such as global food security, malnutrition, high food prices, depleted soil fertility, and many more. With an increased amount of land, we would be able to overcome these challenges, but how would we make this land useful? Seed germination is the very first stage in plant growth, and it is the beginning of the growth and development of the seed. Understanding what affects the growth of the plant is vital to prevent any factors from affecting maximum seed germination. One of the major stressors that inhibit plant germination is sodium chloride. Increased salinity within an irrigation system decreases crop yield of a region. Having excessive sodium chloride in agricultural land or irrigation systems hinders the growth of seeds. What is a bioassay test? A bioassay test is a test which is used to understand the effects of different concentration of a substance or chemical within a controlled laboratory setting. Bioassay tests are vital to understanding the effects of sodium chloride on plant growth as it allows the experiment to be quantifiable. Understanding the basics of plant growth has the potential to leading to many innovations in plant physiology and a thriving environment. The main point of this project is to understand the effects of sodium chloride on seed germination, specifically on the model organism used, Lactica stavis, also known as lettuce. This experiment aims to understand the growth of embryonic roots under the influence of different concentrations of sodium chloride. This experiment has significant real world applications contributing to the optimization of irrigation systems, crop development, policy formulation, and land usage planning. It aids in optimizing irrigation by avoiding saline concentrated areas, assists in crop selection based on salinity tolerance, and develops strategies for environmental conservation in high salinity regions. Additionally, it supports policy development by providing insights and preventive measures to reduce the risks of plant being placed in areas with high salinity. This project can lead to many advancements in many fields and can be explored in many ways, such as investigating economic implication, studying the roles of soil microorganisms, delving into genetic modification, and more. Further research could assess the benefits of implementing measures and guidelines, understanding the advantages of policy regulation on agriculture, and exploring the impacts of microorganisms in saline soil. It has the potential to deepen our understanding of sodium chloride's effects on seed germination and contribute to the discoveries in gene modification for enhanced resilience to sodium chloride and its impact on plant growth. These implications underscore the research's potential to provide valuable insight for a sustainable agricultural environment. The impacts of researching the effects of salinity and embryonic root germination significantly outweigh the negative aspects. Understanding this topic can lead to many developments in various fields, as well as deepen our knowledge on fundamental biological processes. The questions that will be answered in this project are: at what concentration does sodium chloride become toxic to seeds? And is a certain amount of sodium chloride beneficial to seed germination? I hypothesize that the higher the concentration of sodium chloride, the less radical germination there will be found. I believe that no amount of sodium chloride will be beneficial to the growth of Lactica stavis and will only have negative impacts. The thing that I would be changing in this experiment is the concentration of sodium chloride given to the seeds. The aspect of this experiment that would change is the growth in millimeters. And some of the variables I would keep constant in this experiment are the temperature, location, seed packet, amount of water, and the same salt. Here are the materials I used. During the experiment, I started off by gathering all of the necessary materials and placing a sterile weighing boat onto the weighing scale. I zeroed out the scale to only consider the weight of the sodium chloride. I then weighed out 11.69 grams of salt onto the weighing boat, which I poured into 1,000 mLs of distilled water. I mixed the solution thoroughly until no salt could be seen in the solution. I then poured out 100 mLs of the zero point 0.2 mol solution and set aside for later use. I repeated this process for each of the solutions, adding more distilled water each time to decrease the salinity. I then cut out 4 circles out of filter paper to the measurements of the petri dishes to ensure that when the concentrations were given to the seeds, they would absorb the solution. After cutting out the circles, I placed them onto a petri dish and labeled the lids for each petri dishes. Using a pipette, I measured out 2 mLs of each solution in the corresponding petri dish. I then carefully extracted 8 seeds from the packet of seeds without crushing them. I evenly spaced them out throughout the petri dish. I then closed them all up and placed them in a Ziploc bag to ensure that the seeds absorbed the moisture and left it in a dark and enclosed environment for a 5-day incubation period. After the 5-day period, I measured out the radical length of each seed and recorded all of the data. The first image is the labeled petri dish after the 5-day growth period. The second is when I was extracting the seeds for measurement, and the last image is preparing the seeds for measurements. As you can see by the data, 0.2 moles had no growth, while the control variable and 0.05 moles had significantly more embryonic root growth. The results can also be exemplified in this graph shown below. In this experiment, 4 different sodium chloride solutions were utilized. Control, which had no sodium chloride, 0.5 moles, 0.1 moles, and 0.2 moles of sodium chloride. The petri dish with 0.2 moles showed no growth, likely due to the high concentration, while 0.05 moles exhibited substantial embryonic root germination. The experiment's results indicated that the lower the concentration of sodium chloride in water corresponded to greater embryonic root germination. The highest germination, excluding the control, occurred at the lowest concentration of 0.05 moles of sodium chloride. Conversely, the highest concentration of 0.2 moles of sodium chloride resulted in no seed growth. The control petri dish without sodium chloride exhibited significant growth compared to the others. The initial hypothesis is confirmed by the observed results of this experiment. This project holds significant real-world applications, contributing to the optimization of irrigation system, crop knowledge development, policy formulation, and land usage planning. It aids in the optimizing irrigation systems by utilizing the project's findings to avoid areas with high salinity concentration. Additionally, it assists in crop selection by enhancing knowledge and strategically placing crops based on their salinity tolerance. This project addresses environmental concerns by developing strategies to preserve plant life in high salinity areas and minimizing the environmental impacts of sodium chloride. It can also contribute to policy development by assisting policymakers in formulating effective guidelines and preventive measures to reduce the risks of damage to plant life. These are some of the resources I used when completing the experiment and completing my research. These are the rest of my citations. Thank you for listening to my presentation.